Hi everyone, this is Sherelle Hadrick and I will be discussing factors that influence fat deposition. Before I discuss the factors affecting how fat is deposited in the body, I'd like to provide a simplified diagram summarizing the general process of fat cell growth and proliferation. During hyperplasia, or the increase in number of fat cells, three preadipocytes were formed from the initial cell. When energy intake exceeds energy expenditure, fat storage increases, causing fat cell hypertrophy or the growth of a fat cell. After continued excess energy intake, these cells reach maximum hypertrophy and undergo hyperplasia again. It is important to keep in mind that even with fat loss through energy restriction and exercise, the size of fat cells can decrease, but the number of fat cells will not. A handful of enzymes, receptors, and proteins affect fat cell size and proliferation. Some of these include adrenergic beta receptors, adrenergic alpha receptors, lipoprotein lipase, and acylation stimulating protein. Beta receptors stimulate lipolysis, or the breakdown of fat, and decrease the size of fat cells, while alpha receptors promote the storage of fat and increase the size of fat cells. Lipoprotein lipase is an enzyme that acts as a gatekeeper by promoting the storage of triglycerides in fat cells and increasing fat cell size. The presence of insulin, prolonged fasting, and exercise has been shown to increase lipoprotein lipase function. Lastly, acylation stimulating protein also promotes triglyceride storage in fat cells and may have an increased presence in obese individuals. Sex hormones greatly influence where fat is deposited. The deposition of adipose tissue can be broadly categorized into two patterns. The lower body fat pattern, also known as peripheral fat, contributing to the classic pear-shaped figure, and the upper body fat pattern, known as visceral fat, contributing to the classic apple-shaped figure. Generally speaking, women are more inclined to have a pear-shaped fat pattern, with fat primarily stored in their hips and their thighs, while men are more inclined to have an apple-shaped fat pattern with larger abdominal deposits. This is due to a difference in primary sex hormones. Subcutaneous lower body fat cells have a larger quantity of estrogen receptors, while visceral fat cells have more testosterone receptors. Subcutaneous fat cells have more alpha receptors that promote fat storage and make breaking these fat deposits down difficult. Central fat cells tend to have more beta receptors that stimulate lipolysis or fat breakdown, making these fat deposits more inclined to shrink with proper diet and exercise. The hormone cortisol is another factor that influences fat deposition. A major study conducted in 1997 found that increased intra-abdominal fat deposition occurred in patients with major depressive illness. Cortisol is secreted by the adrenal cortex in response to the stimulus of stress and stimulates a rise in blood glucose levels that release fat from stores. Visceral abdominal adipocytes have a cortisol sensitivity, which results in an increase in lipoprotein lipase activity in the abdominal region. The factors mentioned in this presentation merely scratch the surface of all of the factors influencing where and how fat is deposited. However, time constraints prevent me from further discussing them. This concludes my presentation. Thank you.